Welcome all. I'm Dr. Charlie Roberts, Cancer Center Director here at St. Jude Children's Research Hospital. And it's really both my pleasure and my honor to introduce Dr. Kim Stegmeyer as this week's presenter. I've known Dr. Stegmeyer for many years. She's a colleague, a collaborator, a friend. But really more important than that, for today's purposes, she is a true leader in the field of discovery of new cancer targets and therapeutic leads in childhood cancers. While it's common for research laboratories to focus on a specific disease, Dr. Stegmeyer instead has focused on discovery of opportunities for therapeutic intervention, finding vulnerabilities across a wide variety of pediatric cancer types that she can then pursue mechanistic understanding and, importantly, therapeutic translation. And this is something that her lab does exceptionally well and for which she has been quite successful. She continues to be active in other roles. She is a physician scientist and attends on the inpatient service at Boston Children's Hospital several weeks a year. She's rapidly risen through the ranks at Harvard, and in 2018, she was promoted to the rank of full professor at Harvard Medical School. She's currently chair of the AACR Pediatric Cancer Working Group. She also serves as vice chair of the Scientific Advisory Board for St. Jude Children's Research Hospital, and she served as chair of the NIH Moonshot Fusonc 2 Steering Committee. She is the recipient of many awards, including an NCI Outstanding Investigator Award, E. Johnson Award for Research in Pediatrics, Stand Up to Cancer Innovative Research Grant awardee, and new member Outstanding Science Award from American Pediatric Society. On a personal note, Kim is one of the most thoughtful, dedicated mentors and collaborators that I can think about. It's always a pleasure to collaborate with Kim. And trainees truly flock to her lab to be in her lab, and because of her mentorship. That's not an overstatement. She's really become a hub of trainees and mentorship in her science, both because of the quality of her science, but the quality of her mentorship as well. I don't know where she finds the time, but she's also a highly accomplished ballerina, and I think continues to dance in addition to all of her scientific impact. With that, Dr. Stegmeyer.
Charlie, thank you so much for that incredibly generous introduction. It's a real honor and pleasure to be able to speak here today. So I'm going to be presenting on a project known as the First Generation Pediatric Cancer Dependency Map. So just a few disclosures, but none of them directly relevant to this work. So I think we would all on this call agree that there has been absolutely tremendous progress made in treating children with cancer over the last several decades, but we're still left with some very sobering facts despite this progress. So cancer is still the leading cause of death by disease in children between the ages of 1 and 14 in developed countries. And for even the survivors, we see that they face significant morbidity and cumulative excess mortality. So we have a lot more work to do in the treatment of children with cancer. So there are a number of cancers that represent an unmet clinical need. I just showed 3 of those here: acute myeloid leukemia, high-risk neuroblastoma, and patients particularly with metastatic Ewing sarcoma, where really nothing has improved their outcome for many, many years. And so as Charlie told you, our lab works on a number of different diseases. We've been focused on leukemia, Ewing, and neuroblastoma, and really using different types of omics approaches, including chemical genomics and functional genomics, to try to better understand the mechanisms driving these diseases, to find new targets, and then ultimately new therapies for these children. And so one of the paradigms that we have all been following in the last several years is this idea of molecularly informed therapy for pediatric cancer, that we can profile tumors, their genomics, their RNA sequencing patterns, their proteomics perhaps, and identify new targets, match them to targeted therapies, and then hope to see very durable responses, as has been the case for incorporating imatinib to target BCR-ABL in children that have BCR-ABL positive ALL, where really this has transformed their care, most of these children will no longer need a bone marrow transplant. And so now, a number of years ago, we decided to try to really look at this approach more deeply for children with leukemia. And I was very fortunate that Jana Pickman, who at the time was a pediatric oncology trainee in the lab, was up for this challenge. And I'm really excited to say Jana now has her own lab at the Dana-Farber. And so we formed a consortium known as the Pediatric LEAP Consortium for Leukemia Precision-Based Therapy. And the idea was that we could readily sample leukemia from children with high-risk refractory leukemias, relapsed leukemias, and then perform genomic tests. And in this case, we used clinical grade genomic tests. So one was approximately 100 gene panel. And then we also incorporated tests that were evaluating for fusions. And then in a collaboration with Jeff Tyner's lab, we also performed small molecule screening using patient leukemia samples treated in vitro. And the idea, of course, was to identify targeted drugs for these children, and then when possible, to incorporate those drugs into their therapy. And so this was really a feasibility study to say, is this feasible to identify actionable alterations with a matched targeted therapy for children with leukemia? And it began as a meeting in 2014. We invited 6 institutions, and then we're really fortunate that the St. Baldrick's Foundation funded our consortium. And then from July of 2016 to now, the LEAP Consortium expanded to 15 institutions. And we found that the enrollment was so robust and the demand so high that we've actually expanded the enrollment now twice. And so just from a bird's eye view, what do we see? We see a whole collection of mutations in the leukemias from these children that really do largely map what has been reported. We do find that there are activating mutations in kinases, which are readily druggable. We also see fusions as another important theme, as you would expect in pediatric leukemia. And particularly RAS pathway mutations are quite common. And so what we did is we actually tiered these mutations based on our confidence in matching the mutation with a targeted therapy. And we found that about 18% of patients had a high confidence match, meaning that there's actual clinical data for that mutation and response in patients. And then we found that close to 80% had a match of any tier. And I want to share with you one vignette of a child who really had a dramatic response, and to point out that that child actually was not in the highest tiers in terms of their match. It was a tier 3, meaning there wasn't yet good clinical data for that match. And so this was a 10-year-old girl who had B-ALL. She was diagnosed in 2016, and she had treated with standard chemo and then relapsed, and salvaged chemo, and she relapsed. She had a bone marrow transplant and relapsed, and then had CAR T and relapsed. And when she came to the Farber, she enrolled on the LEAP study, and she had fusion testing ultimately done that revealed an RCSD1-ABL2 fusion. And now you would predict that this fusion would be responsive to the drug imatinib. So at the time, she was not doing well. She was not responding to cytotoxic chemotherapy, and actually had multiple infections. She went on imatinib and went into a complete remission. And she was in a stable remission. She had another transplant, and she's been in a remission for over 2 years now on imatinib. So for this trial, this was really transformative. And I think it's important to know that it's not only in the case of children who have a match to targeted therapy where there's benefit. We've had some children where their diagnosis was changed based on the genomic data. We've had other children where a germline predisposition was revealed based on the genomic data, and that then allowed family members to be tested, and those patients and their families will have earlier screening for secondary cancers that they might get as adults. And so I just want to pause just to thank the many people who participated. As I said, now 15 institutions are involved, and this has been an incredible collaboration with our colleagues across the country. One of the things, though, that I think this study reveals, as well as a number of the other studies that have focused more on children with solid tumors, is that probably at best, 20 to 30% of children have a clinically actionable mutation, given our understanding of the genomics and our armamentarium of targeted drugs. But that means that a lot of children don't yet have a good match targeted therapy. And in fact, we know that in the case of pediatric cancers, many of the targets are difficult drug targets. They involve fusions that involve transcription factors, and it is notoriously difficult to make drugs that target transcription factors, although I will say not impossible, and many of us are optimistic about new approaches in chemistry to tackle that problem. And so over the last several years, a number of important themes have emerged in pediatric cancers. One is that fusions are common. These are typically an initiating event, and they're required for tumor maintenance. And we see these fusions across the spectrums of cancers: leukemias, solid tumors, and brain tumors. Often the fusion involves a transcription factor or a chromatin regulator, but when they've involved kinases, that is where we've seen very dramatic responses to targeted therapies. And again, across the spectrum of diseases, from leukemia to solid tumors to brain tumors. Pediatric cancers are now known to generally have simpler genomes compared to adults, with few recurrent point mutations. This is very nicely shown here in this image from Stefan Pfister and colleagues, where you can see on the left are pediatric cancers with a much lower coding mutation per megabase compared to the adult cancers. These tend to occur in very specific developmental states, and they typically are seen only in children. And unfortunately, they also have not been terribly responsive to immune checkpoint inhibitors. And so what I'd like to tell you about now is first our efforts around the development of a pediatric cancer dependency map. And then I'd like to highlight two really interesting targets that have emerged from this data. One is a target known as TRIM8, and the second is a target known as NXT1. And so what's the idea of a cancer dependency map? This is work that was started now many years ago and really spearheaded by Bill Hahn and the Broad Institute. And it was the idea that we could take collections of cancer models, in this case, largely cancer cell lines, and profile their molecular features, and then perturb them very systematically, either with small molecules or with genetic perturbations, and then bring all that data together and, using computational approaches, highlight new candidate therapeutic hypotheses. And so I'm going to focus the work most recently that's been using CRISPR-Cas9. And as in model systems, where there are pros and cons to each model system, there are also pros and cons to each screening modality. And I think CRISPR has a number of advantages, particularly over RNAi. And one very important one is that there is reduced off-target effect. Another is that you tend to get very strong target depletion. And now there are very good rules around guide design. But there are some disadvantages that are important to know about. One is that when you make cuts in DNA using CRISPR-Cas9, that is deleterious. You're causing some DNA damage. And if you have regions of amplification and you're making a lot of cuts, that can actually be toxic. And so the Broad has developed algorithms to try to correct for cutting toxicity in regions of amplification. There's also a lack of a dose response, right? So you either generate a heterozygote or a homozygote deletion. And some models we just have not been able to screen. They don't seem to tolerate Cas9. And so this is how these screens tend to work. We've been focusing on the Broad Institute Avana library. This library has 7,000 CRISPR guides. It targets 18,000 genes with 4 unique guides per gene, and over 700 cancer cell lines have been screened. So the cancer cell lines are rendered Cas9 positive and activities confirmed, and then they're infected with the library. They're selected with puromycin, and then they're passaged usually for 21 days. And then genomic DNA is harvested at the end of that time. And then the plasmid pool and the genomic DNA is amplified. And then you have this particular sample sequenced. And so we've been most interested in those guides that deplete, as that may represent genes that the cells are dependent on for growth over time. So, the Broad Institute initially was very focused on building out the DepMap and largely focused on adult cancers. And a number of years ago, Charlie Roberts, Tal Golub, and I had a talk about trying to really expand and highlight now, rather than just adult tumors, pediatric cancers, and create a pediatric cancer dependency map. And so this has been an incredibly fruitful effort over the years, and we were very fortunate that the Broad Institute and Dr. Golub and Dr. Hahn really enabled us to do this project. And so this has been a terrific collaboration with many people over the years. And so this data is made public. It's made public approximately 4 months after the data is generated. And you can see here the representation of pediatric cancer models currently in the data. And this first build of the data was actually led from a computational perspective by Nikesh Daria, who is another really talented pediatric oncologist and computer scientist who trained in the laboratory. And so I do also want to thank, again, this project was a tour de force with many people involved, but to highlight Todd Golub and Bill Hahn, as well as Paquita Vasquez, who is our project manager. And then Charlie Roberts, who really was one of the leaders getting this off the ground. And then a number of younger investigators who have been disease champions as we have built this project out. And so the first question, of course, was, well, how well do these pediatric cancer cell lines map to primary tumors? You know, we often get criticized that, well, cell lines, are they really representative of primary human disease? And so what Nikesh first did is he looked at RNA sequencing data from the cell lines and compared it to primary tumors. And he regressed out from the primary tumor data immune signal. And so what you can see in this UMAP projection is some of these cell lines like Ewing sarcoma, neuroblastoma, and rhabdomyosarcoma. They map very well to the primary tumors. And so there are many different types of questions you can ask about dependencies. You might say, well, is there a lineage dependency, solid tumor versus hematopoietic? Or is there a cancer type dependency like AML versus ALL? Or genotype dependencies, either within the disease or across diseases? There was a paper that was published in 2017 by the Broad that also looked at other types of dependencies. And so one classification of dependencies are thought to be mutation driven. So if you have, for example, AML that has a FLT3 mutation, you would predict that those cell line models would be more dependent on FLT3. Another example are expression-driven dependencies. And so we often see this with lineage-related transcription factors, and I'll show you an example of that later in the talk. There can be cyclops dependencies where there's a copy number event in one copy of the gene, rendering dependency on the remaining copy. And then another type of dependency called a paralog dependency, and I'll highlight an example of this, where if you have paralogs, if you lose one member of the paralog class, you may be more dependent on the other. And so just giving you some examples of how we look at the data. Here are 3 different genes. On the x-axis is the gene effect score, and each of these hash marks is a cell line. So the more negative the gene effect score, the more dependent that model is on that gene. And yet you can see here this gene, which is an olfactory receptor, the distribution of the curve is basically centered around 0, meaning that cell line models are not dependent on the olfactory receptor for growth over the 21-day period. Whereas TOP2A, which is a targeted chemotherapy, this is what we call a pan-lethal, meaning that all of the models are highly dependent. And we usually use less than -0.5 as a cutoff for calling a dependency. Versus ISL1, which is what we call a selective dependency, meaning that most of the models are centered around 0, but you see this nice tail where there are several models that are highly dependent on ISL1. And ISL1 is now known to be a member of the core regulatory circuitry in neuroblastoma, and most of these models are in fact neuroblastoma models. And so one of the fundamental questions that we wanted to address with this data, and we focused our first big data analysis on the solid tumor and the brain tumor data. And the first question was, well, you know, pediatric cancers, they don't have that many mutations. Might that then translate to fewer dependencies? And what we found is the answer to that question was no, that in fact, pediatric cancers have as many selective dependencies as adult cancers. And by selective, we mean that that's a dependency in that disease against all the other hundreds of models that have been screened. And so what you can see here in color are the different pediatric cancers, and you can see that their number of dependencies on the y-axis is really scattered throughout the collection of adult cancers. And so, of course, one of the things that you can do with the data is to ask, well, might we be able to drug repurpose? So are there genes that score in specific diseases against which we already have drugs? And so this is just highlighting a few of those examples where the neuroblastoma models that have either ALK mutations or amplification are ALK dependent. And for BRAF dependency, again, it's the models that tend to have BRAF events. Versus, for example, something we found very interesting is that a number of the pediatric solid tumor models are actually MCL1 dependent. And so Lily Gunther in the lab went on to validate that particular finding, which we thought was quite interesting, as there are MCL1 inhibitors now in early phase studies in leukemias. And so one of the other questions, of course, we were interested in is whether pediatric cancers express and have unique dependencies. And so one of the things that we found is a number of these cancers had unique dependencies that were associated with gene expression. And one of these that I highlight is neuroblastoma, where we found that these neuroblastoma cell lines were highly and uniquely, in some cases, dependent on some key transcription factors, such as ISL1, HAN2, MYC-N, and FOX2B. And in work that was a great collaboration with Tom Look's laboratory, as well as Rick Young's, with Adam Durbin, who's now on faculty at St. Jude, leading the project with Mark Zimmerman and Nikesh Daria, really identified these strong dependencies as members of a core regulatory circuitry. And there have been a number of other laboratories in Europe who came out with some outstanding papers, also published in Nature Genetics, really defining this key regulatory circuitry in this disease. And so, of course, we were also interested in whether there were some unique dependencies that were not previously on our radar. And this is one example where in Ewing sarcoma, we saw this incredibly strong and unique dependency on TRIM8. And this score of TRIM8 was not just in the Ivana screen, but I'm going to show you this scored in a number of different CRISPR screens. And so we got very excited about thinking more about TRIM8 in Ewing. And so I'd like to tell you first a little bit more about Ewing and then really drill down on the data focused on TRIM8. So Ewing is the second most common pediatric bone cancer. It's treated with very intensive chemotherapy, surgery, and radiation. And as you saw before in those survival curves, about 70% of patients with local disease will survive. But for patients with metastatic disease or relapse disease, the survival is very poor. This is in many ways a canonical pediatric cancer. It's driven by a fusion, most typically an EWS-FLI fusion. EWS is a RNA binding protein, while FLI is an ETS transcription factor. And the fusion involves the strong transcriptional activation domain of EWS fused to the DNA binding domain of FLI. And there are a number of other more rare fusions. The second most common is EWS-ERG. Very interesting disease in that the cell of origin is not certain. It's thought to maybe be a mesenchymal stem cell. A lot of work has been done by a number of investigators over the years that has taught us that EWS-FLI acts as a pioneer transcription factor. It recruits the BAF complex and opens up chromatin. And EWS-FLI then binds at GGAA repeats, leading to an altered gene expression program. And so one of the efforts of our lab a number of years ago was to ask, are there recurrent mutations in Ewing? And what we found here is that, in fact, recurrent mutations are quite few. This was work led by Brian Crompton in the lab. There's also a very important paper that came out of St. Jude in work led by Mike Dyer and Olivia Delatra, as well as work that came out of the NCI led by Javed Khan that really all showed a very similar thing: quiet genomes, few recurrent mutations. And so we got really excited about thinking more deeply about how we might target EWS-FLI. And we're really fortunate to be a part of the NCI Cancer Moonshot Fusion Oncoprotein and Childhood Cancers Consortium. And so I'm going to tell you a little bit more about that work and how it intersected with the dependency map. And so one of the projects as part of our grant was to actually ask what regulates EWS-FLI, and can we begin to make reporter cell line models and use those both in CRISPR-based screens, but also in small molecule screens. And so what we did is to generate an EWS-FLI GFP mCherry expressing 293 reporter model. And so these cells express EWS-FLI GFP, and they also express mCherry. And the mCherry can be used as a control to rule out targets that may generally perturb transcription or translation. And so here, what was done is this is some pilot experiments to show that if we take this model and we delete with CRISPR EWS fly. We see loss of GFP signaling here based on Western blot. And by flow cytometry, we can see a very nice shift in the curve where we now have this population of GFP low, meaning EWS fly low, but cells that still maintain their M. cherry levels. And so Alex Song, who's another really talented postdoc, has been leading our work focused on these types of screens in Ewing and TRIM8. And so what Alex did is after he generated that model, he then took the model, infected with a CRISPR library, and now used the Ivana library from the Broad. He then did flow sorting for GFP low cells or GFP high cells, and then the guides were sequenced after amplification. And so we got really excited when the Broad called us to say, hey, this target TRIM8 is scoring. What do you think about TRIM8? And we were, of course, extremely excited because we knew that TRIM8 was also scoring in the dependency map. And so TRIM8, when deleted, was scoring in this GFP high population. And so what's TRIM8? It's an E3 ubiquitin protein ligase. It's been variably reported as a tumor suppressor in some contexts and an oncogene in others. It has a ring domain. It has 2 B-box domains and does have a nuclear localization signal. And so this just shows you how the different lines performed in the pediatric dependency map data, which is intersected with all of the adult data. It's integrated as one data set. And so here what you can see is that it's not just the EWS fly models, but also the EWS ERG model is scoring in this particular screen. And only these models are scoring. None of the other cancer cell line models that were screened scored. And so one of the first things that we need to do is actually validate the dependency. And so here what Alex did is he went at low throughput and knocked out TRIM8, and then showed that he got good knockout by Western blot, and then showed that in all the models that he tested, the loss of TRIM8 was deleterious to growth of the cells. We wanted to make sure that this wasn't just an in vitro phenomena, so Alex then took those cells and looked at their performance in vivo, and similarly found that deletion of TRIM8 impaired growth in vivo. And so the prediction from the 293 screen is that when you lose TRIM8, that EWS fly would go up, which is a bit paradoxical in light of EWS fly functioning as an oncogene. But that's what we saw, is when you delete TRIM8, E-fly goes up, as does a direct target of EWS fly, NKX22. We also saw the same in an EWS ERG line, deletion of TRIM8, increase EWS ERG, increase a direct target also of EWS ERG, which is NKX22. And importantly, this wasn't just a CRISPR phenomena. When Alex used siRNA, he found the same thing, that EWS fly similarly went up. And so then the other hypothesis is, well, now if you overexpress TRIM8, if TRIM8 is regulating EWS fly, you'd expect EWS fly to go down. And that's actually what Alex saw. So he overexpressed TRIM8, EWS fly goes down, the target goes down. And as was predicted and validated in many other studies, the growth of the cells is impaired. And then he also used a dominant negative TRIM8 that lacks the ring domain. And here again, E-fly goes up, the target goes up, and cells don't grow. And so I'm going to just, in the interest of time, really focus on a few key pieces of data that speak to the fact that EWS fly is a neomorphic substrate of TRIM8, meaning that wild-type EWS and wild-type fly do not appear to be regulated by TRIM8. And so here, when TRIM8's CRISPR deleted, wild-type EWS levels don't change. And in AML cell lines that express wild-type fly, when you either express TRIM8 or you use the dominant negative, there isn't a change in wild-type fly levels. As part of the Fusion Oncoprotein Moonshot Consortium, we were very fortunate to have the opportunity to collaborate with one of the other projects, which is being led by Steve Lesnick's lab, but also Robert Tijan and Xavier Darjak. And working closely with Shasha Chung, who was a postdoc in the lab at the time, Shasha is now running her own lab, we were able to look at whether TRIM8 co-localized with EWS fly in Ewing sarcoma cells. And they had established an EWS fly halo tag model. We then also expressed the TRIM8 GFP and found very nice concordance of localization of both TRIM8 GFP and EWS Fly Halo. And so another system that our lab really has become fond of using to validate targets is a degradation system. And what this allows you to do is to acutely degrade your target of interest and really look at very early time points after degradation. And so how does the system work? So it's a DTAG system, and a DTAG is a heterobifunctional molecule that binds specifically to a mutant FKBP12 and then an E3 ligase to induce proteasomal degradation of your target protein. So the strategy that we've been using is to concurrently knock out the endogenous target, in this case TRIM8, while expressing a tagged target. So this target protein would be TRIM8. It would have the FKBP mutant tag. When we treat with the DTAG molecule, it binds the DTAG and also binds to an E3, either cereblon or, in the case of the work that I'm going to show you, VHL, recruits the E3 machinery, and then you get targeted protein degradation. So Alex very successfully established DTAG models for TRIM8. He looked at both N-terminal tagged and C-terminal tagged, and found that with both of these approaches, when we treated with a DTAG that recruits VHL, he got very nice degradation of TRIM8. EWS Fly goes up, NKX2 goes up, and there's a very profound effect on growth, completely concordant with what we saw with CRISPR and RNAi. And so the beauty, though, of this is that Alex could then do global proteomics at a very early time point. And so he looked at global proteomics after 6 hours of degrading TRIM8. And what he found is that EWS Fly was one of only a very small number of targets whose total protein levels actually increased with the degradation of TRIM8. So now we have this interesting paradox where when TRIM8 is repressed, EWS Fly goes up. When TRIM8 is repressed, growth goes down. And so the question, of course, was, is the increase in E-Fly mediating the impaired growth? And so the first thing Alex did is a very simple experiment where he just introduced exogenous EWS Fly and said, well, if we express more E-Fly, is that deleterious? What he found is that the cells did not grow as well, and we also saw evidence of cell death with induction of cleave-PARP and cleave-caspase-3. He also looked at this in vivo in a patient-derived xenograft model and found that overexpression of E-Fly in that xenograft similarly impaired growth in vivo. And then he did a series of very important rescue experiments, and I'm going to highlight one here, but we saw very concordant results across them. So Alex had also developed, as part of a collaboration with Nathaniel Gray's lab, a DTAG Fly model that allows him to degrade EWS Fly. And so when you degrade EWS Fly, you see a deleterious effect on growth, as you would expect. So then what he did is he concurrently CRISPR deleted TRIM8, EWS Fly goes up, TRIM8 is gone, there's a deleterious effect on growth. And then he could titrate back with the DTAG molecule the amount of EWS Fly in the cell back to baseline. And when he did that, he saw full rescue of the loss of TRIM8. So we think that this is really a Goldilocks phenomenon where in Ewing sarcoma, you need to tightly control EWS Fly. And we know that EWS Fly is very toxic to most cellular contexts. And it was actually very difficult to make the 293 model. And so EWS Fly, we think, is a neomorphic substrate of TRIM8. There's a lot of data. In the interest of time, I did not show today. TRIM8 deletion was selectively toxic to only Ewing compared to over 700 cancer cell lines. And we think this might be an interesting approach in other fusion cancers that we're going to begin to explore informatically. So I'm going to end with one other new vignette. And this is a project led by Claire Malone, another outstanding postdoc who's been training in the lab. And what Claire got interested in is saying, well, you know, a lot of the therapy that we use to treat children with cancer that's been effective is really cytotoxic chemo that leads to cell death. And so her question was, can we find a target that when deleted is as toxic as these targets of chemotherapy, but is selective for that particular cancer of interest? And so I alluded to this before, that targets of many of the chemotherapy chemotherapy agents are actually pan-lethal targets in the dependency map. And this just is one example of etoposide. So virtually all the cell lines in the dependency map are dependent on TOP2A. And so Claire wanted to focus this effort on neuroblastoma. And so this is a disease that arises from the developing autonomic nervous system. It's the most common extracranial tumor of childhood, solid tumor, I should say. The cytotoxic chemo is curative for some, but at the cost of a lot of short and long-term toxicity. And for patients with high-risk disease, overall survival still is quite poor. And children with neuroblastoma, about half of them actually present with metastatic disease. One of the biomarkers of poor outcome is amplification of MYC-N, which is associated with high-risk disease. And Claire chose to focus on the MYC-N amplified subset. So what Claire did is she said, you know, we're going to take just the top 200 targets approximately that are selective in neuroblastoma compared to all the other models that have been screened. And we're going to be pretty agnostic to what they do or whether we think they're druggable. And so she then made a secondary library that included CRISPR guides for CRISPR-Cas9, but also CRISPR-I. And so she then did a series of screens where she looked at a time course with the idea that if a target depletes early, then maybe that's a stronger target. She also did a positive selection cell death screen, selecting for Annexin 5 positive cells. And then she did an in vivo screen with the idea that we want to know very early on whether this target is relevant in vivo. And so these are the bird's eye view results of her screen. What she found is that the concordance between CRISPR-I and CRISPR cutting was actually very strong in the case of the screens that she did. But in the case of cell death, not that many targets score as promoting cell death when depleted. You'll see in red the targets that were internal pan-lethal controls, and these score mostly. And then a small number of other genes score. And I want to draw your attention to NXT1, which is in this cloud with the pan-lethals. The in vivo screens were also very interesting. They showed us that by and large, the in vitro correlated well with in vivo. There are some targets that drop out. These tend to be metabolic targets. But again, what we found is that NXT1 scored in vivo as strongly as did the pan-lethal positive controls. And so at the end of all these screens, what really jumped out was this target NXT1. So what is NXT1? NXT1 is a cofactor for NXF1 mediated mRNA nuclear export. So it forms a heterodimer with NXF1 and is involved in shuttling mRNAs to the nuclear pore to the cytoplasm for translation. And it's the final step in export. And so as Alex did, Claire wanted to first validate this target low throughput, which she did very readily, and found that in this case, there was pretty robust induction of cleave-PARP, as would be predicted from the Annexin screen. And as Alex did, she also made a DTAG system. And so here you can see very nice degradation of the target. This system recruits cereblon and induction of PARP and a very profound effect on growth. So Claire also wanted to validate her in vivo findings. And in this case, we were able to establish an inducible CRISPR system. Some cell line models, it's very leaky and we technically cannot use it. In this case, it was not too leaky and was a very successful approach. And here what we found is that after establishing tumors and inducing the knockout of NXT1, we actually saw tumor regression. So again, a very strong target in this disease. And so then Claire also was able to do proteomics at a very early time point using her DTAG system. And here what she found is that, as you would hope, NXT1 was one of the top targets. But she also found, quite interestingly, that NXF1 was very strongly depleted at a protein level. And as I told you, NXT1 actually forms a heterodimer with NXF1. So that then raised a very interesting question: do NXF1 levels really go down when NXT1 is depleted? And she found that by CRISPR, that was also the case, that when NXT1 is knocked out, NXF1 goes down. She saw the same thing in the inducible CRISPR model, and she could reproduce the proteomics data at low throughput where she sees degradation of NXT1, and depletion of NXF1. So that was very interesting to us because when you go back and look at the DepMap data, this is the distribution for NXT1. And you can see this is a target that is not a panlethal, that has a tail, whereas NXF1 is a panlethal. So then what Claire did is she looked back at the neuroblastoma models and said, well, are there any models that are not NXT1 dependent? And she found this handful of models here and focused a lot of her work on the Guyman model. And so she then asked the question, well, in a model that's not dependent on NXT1, what happens to NXF1 levels? And so here what she found is when she depleted NXT1 with CRISPR, we do not see a strong viability effect. And she saw no change in NXF1. She also made a DTAG system for NXT1 in Guyman and saw no effect on viability and no effect on NXF1, despite getting very good degradation of NXT1. So we have this interesting model where NXT1 loss leads to NXF1 instability and impaired mRNA export. But why does NXT1 loss lead to NXF1 loss only in some settings, but not in others? And so Claire then went back and mined the dependency map data and asked, are there any predictors of dependency on NXT1 in that data? And what she found was that there was a very strong biomarker, and that was NXT2. And so when NXT2 expression is low, the cells are dependent on NXT1. And so this is one of those paralog relationships. And so in fact, when she then mined larger primary tumor databases, she found that neuroblastoma is one of the poorest expressing tumors for NXT2. So she then asked some very important questions. And one was, can you rescue the effects of NXT1 depletion in a responsive cell by overexpressing NXT2? And so here she overexpressed NXT2, and she found that she saw very nice rescue of the effects of NXT1 depletion on viability, and she fully rescued the depletion of NXF1. She then asked a question of sufficiency. So she now took the Guyman model that doesn't respond to NXT1 loss, and then she eliminated NXT2. And then she saw that, yes, now NXF1 levels go down, and now the cells have a dramatic response, and cleave-part is induced. And so this is the model. We think this is a really interesting example of a context-specific lethality, where NXT1 loss leads to context-specific NXF1 instability and a very profound effect on inducing cell death. And so here you have cells with low NXT2, there's a profound effect, but if normal NXT2 is present, it can compensate for the NXT1 loss. And so we then went back and said, well, how does NXT1 perform in the rest of the pediatric data? And quite interestingly, when Nikesh did a comparison of all pediatric cancers versus all adult cancers and said, are there any targets that are more enriched in the PD cancers? What we found is that NXT1 was actually the strongest differential dependency across these age-based distributions. And so in fact, there are a number of pediatric cancers that have poor expression of NXT2. And now when you look at the distribution, you see these are the adult cancers, more negative the score, the more dependent the models, versus pediatric. And you can see a nice left shift in the childhood cancer models. And so rhabdomyosarcoma, medulla, and even some of the rhabdoids are dependent on NXT1. And so Claire went on to validate that in rhabdomyosarcoma and saw a very nice dependency in multiple rhabdo lines as predicted from the DepMap data. And so kind of projecting into the future, this is a target that's involved in nuclear export. And one might ask, is there any precedence for targeting a protein that's involved in nuclear export? And there is actually. So selinexor is an FDA-approved drug approved for multiple myeloma that's actually been tested by both the St. Jude program as well as Dana-Farber for children with leukemia. And the target of selinexor is XPO1, also called CREM1, which is involved in nuclear export. This target, though, is a pan-lethal, so all the models are dependent on it, and yet there still is a therapeutic window. So we're hopeful that NXT1, which has a better profile predicted by the DepMap data, potentially may be druggable. And so some conclusions from this. We have used these CRISPR screens to identify a novel target that is selective, but as lethal as some of the pan-essential genes that are targets of current therapy. And NXT1 is that target. The loss of NXT1 leads to loss of its essential binding partner, NXF1, in a very context-specific way. And NXT2 expression is a predictive biomarker of NXT1 dependency. And so looking forward, where are we hoping to go with our second generation pediatric cancer dependency map? And we're very excited. There are ongoing discussions about what this will look like and ongoing discussions with a number of folks here at St. Jude. And so we are discussing now expanding the models screened to have broader representation within and across diseases, evaluating other model systems such as neurospheres and patient-derived cell lines, expanding the omics that have been profiled, particularly interested in epigenetics, and then also interested in expanding chemical screening and then integrating those chemical screens with functional genomic data. And I want to end on one, I think, really exciting example that may foreshadow how powerful this approach can be. And this is work that I'm highlighting from Jiyang Yu and Jun Yang here at St. Jude. They have been doing some small molecule work in primary patient leukemia cells and discovered that a subset of TLL was highly sensitive to dasatinib. And then using a number of different omics approaches, discovered that one of the predictors is that dasatinib was targeting LCK. We had similarly profiled a number of TLL models in the DepMap and found that LCK was the #1 selective dependency in TLL compared to everything else that had been screened. And really had a very fun collaboration with their team in a project that was led out of St. Jude. So I think this is one of what I hope is going to be a number of examples of really exciting opportunities to intersect chemical biology and functional genomics. And so the hope is that someday, like we can do for antibiotics and infections, where we say, okay, this patient has a Staph aureus infection, what are the candidate antibiotics that are most effective, and match those antibiotics to the infectious organism. We hope that we can create a larger cancer dependency map that takes into consideration mutational data from the patient tumor, functional data, as well as chemical data to really come up with more effective, less toxic therapies for the patients that we treat. And so with that, I want to thank you all for your attention. I tried to highlight our collaborators along the way. I want to really thank the many people in our lab who've contributed. I also do want to highlight David Root and the platform, the GPP at the Broad, that helped to generate all of this DepMap data that we have really benefited from greatly, and all of our other collaborators, and certainly our funding. So thank you so much for your attention.
Fantastic talk, Kim. I think beautiful work and beautifully presented. So questions are coming in. For folks that have any questions, just type them into the all panelists in the chat box, and we will share them with Kim. So Dan Williamson says, first, wonderful work. Where oncogenic features are shared across tumor types, for example, MYC amplification in rhabdomyoneuroblastomoma, to what extent are dependencies explicable by either genotype or tumor type? And the basket trials in peds make a lot of sense according to DepMap data.
Yeah. So, I think that is a great question. And the answer, of course, is a little bit, it depends. So there are some examples. I'll use P53 as an example. And Charlie's lab has done a lot of work looking at this, where if you ask agnostic to lineage and you say, are there dependencies that P53 intact tumors possess that P53 mutants don't, you will pull up a beautiful circuitry of targets like MDM2, MDM4, USP7, PPM1D that are involved in the regulation of P53 in that pathway. So there are some examples where, if you didn't know anything about the biology of that target and the mutations, you could really learn an amazing amount from the data, and therapeutically that's really relevant. There are other times where it's more complicated, where we think there are probably layers of biomarkers, where it's probably not sufficient across all of these diseases just to have the one biomarker, that there may be other things that are modulating that particular biomarker. So I think it's sort of a mixed answer. And I think, yes, in terms of your comment about like the match targeted therapy trials through Children's Oncology Group, I think yes. And you'll see if you notice that, for example, BCL2, right, in solid tumors, it scores in our data in neuroblastoma. And that's where in all the prior work, there was interest in BCL2 inhibitors like venetoclax was in neuroblastoma. So I think there the data is quite nicely matched to what people in the community had previously published.
Great. Next question from John Zawitowski. Can you comment on the lack of recurrent mutations, SNVs, in pediatric cancers you are familiar with? Do you feel there is utility in whole genome sequencing versus panel or exome to identify potential promoter, enhancer, pioneer factor changes that might be influential and potentially targetable, either in the germline context or mutation context?
Yeah, so another really great question. And in some ways, this may vary depending, what the right approach is depends a little bit on the disease and also our armamentarium right now, right? So, I think as an example, Ewing sarcoma, right now, when many different groups have done either whole exome sequencing or targeted sequencing, you do not readily discover that many mutational events. There are rare EZH2 activating mutations reported, but you really don't discover that many events that would lead to a targeted therapy, right? And in our own data, the answer would be we didn't see any. But of course, the more you sequence, the more you find. That's right now, though. I think that in the case of STAG2 mutations, right, those are picked up with exome sequencing or panel sequencing, but we can't drug that yet. But if we had a STAG1 inhibitor, right, and so I didn't show you this, but there's a paralog relationship between STAG1 and STAG2. So if STAG2 is gone, the cells are very dependent on STAG1. So at some point in time, if we actually had a STAG1 therapy, then you'd want to know about that STAG2 mutation, right? So I think, I think that it's the complement of approaches. So I think that the panel sequencing or exomes are important. You don't gain the fusion. So the example that I showed you of that patient with the RCSD1 ABL2 fusion, that of course wasn't picked up on the targeted panel. And it was only when we had an adequate fusion panel that that was picked up. So that child actually had 2 rounds of discovery before it was found. So I think it's going to be this complement of being able to find the common point mutations, being able to have approaches that adequately identify fusions, and then this whole new space of non-coding is a really exciting one. And we're very thrilled to be a part of a St. Jude consortium led by Jimwei Zhang that's really starting to look at this. And we're starting to get hints of really exciting examples where that type of genomic event is being discovered in terms of outlier expression in the DepMap data. So I think more to follow. And unfortunately, I think we're going to need complementary sequencing approaches.
Excellent. Next question from Charlie Wright, who says, great talk, exclamation mark. For the goal of new pediatric therapy, do you think that dependencies that are selective in the context of pediatric versus adult are more promising to go after than cancer type X versus all other cancer types?
Yeah. So, another interesting strategy question. So, when we looked at all pediatric cancer versus adult, as you might predict, we didn't find that many, right, dependencies that were unique to all pediatric cancers or even enriched across pediatric cancers. If you saw in that figure, IGF-1R was there, and that was a target that there was a lot of interest in childhood cancers. It hasn't played out as well as we would like. NXT1 was one of the strong targets, and I think importantly, NXT1 is also relevant in some adult cancers. And I think from the perspective of getting interested in the targets, a broader community in terms of the pharmaceutical community, the more applicable the target it is beyond just pediatrics, the more likely people will be interested in a larger drug discovery effort, right? So I think there's kind of the practical and the ideal considerations. But I think, as you saw with TRIM8, it looks like a great target, at least based on this data. It's relevant in Ewing. And I think we need mechanisms to actually focus on targets that are pretty specific just to childhood cancers, right? So, these are things like the fusions that these pediatric cancers have. Interesting targets like TRIM8 that are coming out of these screening efforts. So I think from a practical perspective, the broader the application, the more able you are to get a company to want to think about that target. But I think the reality is some are going to be unique to just peds.
Great. Quick one here. Alex Gout asks, are all the pediatric cell lines used for analysis data generation for PEEP-DEP represented in CCLE?
Yes. The answer to that is yes. They're all in CCLE.
Great. Diana Acevedo asks, again, excellent work. In terms of neuroblastoma, have you evaluated the contribution of tumor microenvironment and how mechanisms of drug resistance might be involved in response to the suggested therapies?
Another great question. Kind of the short answer is no, not really. This is such like the tip of the iceberg. I will say that, the models we used were subcutaneous, right? So that's not the perfect microenvironment, but we were actually pleasantly surprised that if you do what we did, which is to pre-select based on in vitro data, right? So you say, what are the dependencies in vitro and do they hold up in vivo? The answer there was, as you saw, most of them hold up in vivo. What we haven't done is the opposite, is to say we're going to agnostically take 200 targets that we pick based on something, druggables, you name it, and not screen for in vitro. Do we pick up new things in vivo? We actually have not done that experiment to say, are there dependencies engendered by that microenvironment? But I think these are really interesting questions, and the types of things we're thinking about in kind of the second generation is really looking at microenvironment, looking at other types of models beyond just cell lines grown in vitro. But it's a great question, I think really important.
Great. Dr. Stegmeyer will continue to take questions. Given that we're at the top of the hour, I just want to point out that next week's talk in the series, which is the last in the series this spring, will be given by Sigal Kadosh, also in the Department of Pediatric Oncology at Dana-Farber. And I'm happy to announce that due to the success of the series and the incredible buy-in, we'll also be doing a fall version of the series. So next question, the TRIM8 E3 ligase, do you have an idea what its normal mechanism is, what it's doing, what its day job is outside of Ewing sarcoma?
Another really great question. So not based on our own work, but based on the literature, a TRIM8 knockout model was generated, and the knockout of TRIM8 is tolerated. So that's actually encouraging from a therapeutic window perspective. The model did have an immune defect in response to LPS stimulation. So it does seem that at least one of the functions of TRIM8 may be involved in immune regulation. And one of the questions that frequently comes up in Ewing is, well, if you had a molecule, could you look at the interaction of the immune system and Ewing in an immune reconstituted context? And there, the challenges for Ewing, there is no genetically engineered mouse model that's faithful. So I think those studies we won't be able to do in the way you'd ideally want to do them. We could study the impact of the molecule in an immune competent mouse and then study the tumor effects in a patient-derived xenograft or cell line xenograft, but we don't yet have a perfect model for studying immune Ewing interaction. But at least based on the literature, that is one of the putative roles of TRIM8 is really an immune mediation.
Great. Related to this, Natasha Kaplan asks, do you have any insights into whether TRIM8 function has specific roles in mesenchymal stem cells?
Yeah, another really interesting question. So the short answer is no, we haven't looked at that. But what we do know is it's expressed in mesenchymal stem cells. So we've looked at that recently. It actually is pretty ubiquitously expressed in the data that we have in cancer and normal, and it is expressed in mesenchymal stem cells. In the experiments that were done, which are very short time point experiments, where E-fly is introduced into those MSCs, it doesn't look like levels of TRIM8 are directly regulated by E-fly in a short-term context. However, what's very interesting, and I think plays into the Goldilocks phenomena, if you look in well-established Ewing models, like the cell line models, and you look at expression of TRIM8, it's at its lowest in Ewing. And that makes sense, right? Because if there's too much TRIM8, you now are repressing EWS-fly. So you need this like kind of tight control of E-fly and TRIM8 to maintain like what we would say is optimal amounts of EWS-fly. But precisely what it's doing in MSCs, we haven't looked at that yet. We haven't tried to knock it out and study it in MSCs, but I think it's another really good question.
Great. Next question. So relevant to the concordance between in vitro results and in vivo results, you mentioned that they were reasonably similar except for metabolic targets. I'm wondering if can you build on that and talk about how strongly predictive are cell line findings once you go in vivo, because that's obviously been an open question and sometimes an area of concern.
Yeah, no, I think another really great question. We have the data that you saw in neuroblastoma. We have data that I didn't show today in the interest of time where we did a similar type of experiment in AML. We took a collection of, top roughly 200 AML dependencies in one of the earlier builds of the DepMap data, made a secondary library, and then did a CRISPR screen now in 3 models, 2 cell line models, and 1 patient-derived xenograft model that tolerates minimal passaging in vitro. And so, we can infect the cells, they're Cas9, and then re-inject them. We did tail vein injections and looked at marrow compartment and spleen. So this may get at a little bit the prior question about looking at in context, in microenvironment. And very similar to what we saw in neuroblastoma, that most of the time there was a strong correlation between in vitro and in vivo. Marrow and spleen were very highly correlated. There were, again, a few targets that drop out. Some of them are metabolic. Some of the metabolic targets held up very well. So it's not like they all drop out. I don't think, though, yet we have the rules around why things do or don't drop out. I will point out what's very interesting is in the neuroblastoma data, one of the targets that dropped out is actually a transcription factor. And that was very surprising to us. And we had seen other data presented at a meeting where a different group independently had the same result, that they were validating this target in vitro, it didn't hold up in vivo. And I don't understand that yet. It may be that there's some type of lineage switch in vivo and the model no longer relies on that TF. But I think we don't have enough data to really establish rules. But I think that we have seen now that the PDX data looked as good as the cell line data. So it's not like everything fell apart in the PDX in the case of AML. But I think, we don't know what we're missing by starting in vitro.
Great. There's still many people online, so you can continue to ask questions in the chat box function, sending them to all panelists. Maddie von Mecklenburg brings up a question related to the accessibility and use of PDEP, talking about many of the examples you gave are applicable to therapeutic translation. What about PDEP as a research or mechanistic resource? And importantly, how can you make sure that independent labs can still have access to perform CRISPR data, which is quite expensive to do solo?
Yeah, so some really great questions. And so I think that the data, we've focused on strong dependency targets that we think are potentially druggable, right? They're not the easiest drug targets, though, arguably, right? They're not kinases, for example. But we do have some other projects that are looking at targets that are, I don't think are readily druggable at all, but are teaching us a lot of really interesting mechanistic biology. So, I think that, for example, even the core regulatory circuitry project, those transcription factors are not going to be any easier to drug in general than EWS-fly or McN. But we really learned a lot about what is the circuitry that is underlying neuroblastoma and is important for the ongoing growth of those cells. And, there are a lot of really interesting examples, I think, of mechanistic work coming out. And I know Charlie, would say the same, that his lab has some really interesting projects that maybe less focused on therapy and more focused on using this as a tool to understand biology. In terms of the second question, as I said, one of the, I think, amazing things about the dependency map data, which is inclusive of PDEP. So as I said before, we very intentionally decided to have the pediatric data integrated with the adult data, because it allows you to ask the types of questions that you saw us ask, right? There's a richness in having now over 700 models compared to just 100. And you can start asking deeper questions about biomarkers and genotype specificity as well. The data is made public, so we don't hold the data for a publication. You saw that URL that I showed you it's public. The data is public before we have a chance to like spend years and years and publish on it. And so, you know, anyone can mine that data. I think the question about now you want to do your own CRISPR screen. So, you know, the Broad and the pediatric team is always interested in adding more models to the screen. So I think that's one approach, is if you have models that you're interested in screening and having integrated in the data, that is an opportunity. Now, you know, we ask that we hold to the same rules, which is that the data, you know, if it's part of the project, the data does become public after a few months, once it's vetted for quality and it's, you know, it's integrated into the rest of the data. But that's an approach versus the Broad, you know, is really great at collaborating, and, you know, there are opportunities to potentially collaborate with them and use their libraries for your own work, is another possibility. You know, so I think there are multiple ways to think about these types of screens, and some of, you know, the libraries are available commercially. You can also buy them, right? So not cheap, though. I agree, it's not cheap.
Great. So Tim Shaw says, great talk. I was wondering if the CRISPR screening was designed to hit all isoforms.
The answer is no. So that is, you know, that's another potential issue, you know, that we have not looked at closely. We have some projects that are starting to get at, you know, are there other isoforms that aren't addressed, but it's not 100% perfect. It's a good point.
Next question. Why is NXT2 expression low in neuroblastoma and in children? Any insight?
So another really good question. So you guys can still see my screen?
Yes.
Can I still advance it as a question? I put this in just in case. Yes. Okay. So this is why we think it's low in neuroblastoma. And so what we found is that one, there is DNA methylation at the promoter in the models that have low NXT2. And then it also looks like there's, in the models that have high NXT2, there is another start site. And why these models have turned on that start site, we don't know. But they clearly have activation of that start site, and they express a longer isoform of NXT2. And so we think that these two mechanisms in neuroblastoma are contributing to the either low or high expression of the target. And we've seen this DNA methylation pattern also in primary tumors, and it's correlated with expression. So we don't think it's just some kind of strange cell line thing. But a great question.
Still over 100 people on. We're going to get close to wrapping up soon. What does it mean that the gene that you chose to illustrate as common essentiality, TOP2A, is a target that is successfully exploited by chemotherapy? You talked a bit about this, but how do you think about that?
That's another really interesting question. So, you know, when you do these screens and you decide what you want to follow up on, you make choices, right? So we've made the choice to eliminate targets that are universal dependencies across many cancers, if not all. You could say, well, that maybe you shouldn't do that, right? Because those are the targets of chemotherapy, right? And there is a therapeutic window, albeit at the cost of a lot of toxicity. So, you know, we just with intent decided that we want to try to focus on new targets that we think might have a better therapeutic window than chemotherapy. But that doesn't mean that the targets that are pan-lethals might not be candidate therapeutic targets, right? It's just going to be, I think, a harder development path if there's a lot of toxicity. and this is, I would say, one of the challenges with the CRISPR approach, right? The good thing is the off target is much less than RNAi. The downside is there's no graded response, right? So you can't ask, well, if you reduce TOP2A by 50%, are there models that are more or less dependent? Because it's a relatively binary system. So there are now some activities going on to try to look at approaches such as tunable CRISPR-I to see, could you get a more graded response on these targets or not? And I think that could be interesting. The other thing that is, of course, not included here are normal cells. And that has been a long-standing discussion. You know, what is a normal cell line? Does that even exist? You know, and can you screen that? They don't grow well. And you could imagine if you had a comparator set of normal models that were screened against all these targets, you might be able to say, well, at least TOP2A has a stronger dependency across all cancer compared to all normal, but we just don't know yet how to best do that. There's some efforts at the Broad to look at organoid screening, but it's still pretty early. But I think, you know, you're right. Some of these things we're throwing out could be potential targets, and there may be a therapeutic window on cancer compared to normal.
Excellent. Bartholomew Dio asks, I think relevant to the early part of your talk, was the ABLE2 fusion detectable at diagnosis, or was it selected by the different treatments the patient received?
That's another very good question. And my gut is that it was there at diagnosis, but I don't have data. Because that patient wasn't treated at our institution, we didn't have a sample from diagnosis, but a really good question.
Great. So with that, we will wrap. So I want to thank again Dr. Stegmeyer for a fantastic talk, a really engaged audience. You brought it in, Kim. So that was wonderful. Thanks all, and we'll see you next week when Dr. Sigal Kadosh is presenting.
Thank you, everyone, for your great questions.
Bye-bye.